Good morning, everyone. Today, my topic is polyelectrolyte. Polyelectrolyte are the polymer who representing unit barrier of the electron group. These can dissociate in aqueous solution, giving a poly chain charge that make them water soluble. The charge nature of the polyelectrolysis lead to the unique interaction within a counterneous other change molecular and solvent, giving rise to a wide array of the application in the scientist and industry. Now let's talk about the key concept of polyelectrolytes. Firstly, we'll talk about the types. First is cathodic polyelectrolysis. These consist positive charge group such as quarterly ammonium group. Example, pol amino polyelectrolysis. These contain negative charge group like carbohydrate or sulfonate group. Example, sodium polyarychinate. Amyloside polyelectrolysis. These possess both positive and negative charges group on the same chain. Example, protein. And the other one is neutral polyelectrolysis. These have isolated group but are overall charged in a neutral under a specific condition. Now let's discuss about the charge dissociation. Polyelectrolytes iodized in an aqueous solution dissociate into a polymer chain and a free quaternion. This degree of isolation degree on the pH and ionic strength of the solution. The ionic nature providing significant hydration, enhancing water solubility. Now let's talk about the quaternion condition. When the polyelectrolytic dissociate quaternion, opposite charge to the polymeric change are released, supporting the polymer change and partially neutralizing the charge. The effect is explained by many conditions where highly charged polyelectrolysis exhibit particular partial neurolysis by content countering, influencing their overall behavior. Confusing in the solution, polyelectrolytes can take different conformations based on their environmental. In low ionic strength environment, they adapt as external change conformation due to a repercussion between charge on the polymer backward backbone. There are some other important interaction with other molecules. First, opposite charge specimen. Polyelectrolysis can interact with the opposite charge polyelectrolysis forming complex called polyelectrolyte complex. These are the important in drug delivery, layer by layer assembly and coaching. Surfactants. Polyelectrolytes interact with the ionic surfactants leading to the formation of micelles and the charging in the surface behavior. Protein and cell. Polyelectrolyses interact with the biological macromolecule, making them useful in biological applications like gene therapy, tissue generation, engineering, and drug delivery. Application. Firstly, is water treatment. Uh, Polyelectrolytes are widely used in water treatment processes at the coagulation due to the ability to aggregate supplement partial in the neutralizing their charge. Biomedical. In the drug delivery, polyelectrolytes form nanoparticle or the carrying them can enlarge drug protein and then until they reach target. Polyelectric multilayer and many more. Some example of polyelectrolysis are polyacetic acid and salt are widely used in a dispersible and water absorption material. Polystyrene sulfonate (PSS) is a common anionic polyelectrolyte used in electronic application. And the another one is cystosine, a natural cystosine polyelectrolyte derived from a chitin used in biomedical and food industry. Thank you. Now we are going to discuss another topic, principle of protein. The principle of protein is the interdependency field that apply the principle of physics, particularly static mechanism, thermodynamic, molecular dynamic to understand and predict the behavior of protein. Protein are the large complex biomolecule made up of the amino acid which are performing in variety of function within a living organism. The structure and the function are influenced by the physical principles such as energy, force, molecule interaction, and the field focus on a physical property, then governing how protein fold, move, intermediate, and perform their biological function. So, firstly, we are going to discuss some of the concept. First is protein structure. They are firstly primary structure, the sequence of amino acid in polypeptide chain. Secondary structure, located folding patterns such as alpha helix and the beta sheet, stratifying the hydrogen bond. Tertiary structure, it is a 3D shape of the single polypeptide formed by the various interaction by hydrogen bonding, van der Waals force, ion bond, and the disulfur bridge. Quaternary structure, this structure formed by the multiple polypeptide chain subunit coming together. Now let's talk about the protein folding. Protein fold into a specific three-dimensional structure, especially for the function. Protein folding is derived by the physical forms such as hydrophobic, intermediate hydrogen bond, and van der Waals force. Force holding pathway. Understanding how the protein fold help to predict the misfolding, which is linked to a disease like Alzheimer. Now let's discuss about the energy landscape. Protein has an energy landscape that derives the folding and conformation charge. The negative state of the protein corresponded to a lower energy state, free energy. The free energy surface of the protein folding landscape show the pathway between the unfolding and the folding status. Molecular dynamic talks about the protein are not static. They undergo various movements such as conformation, charges, vibration, and rotation. These dimensions are especially for the biological function like enzyme catalysis or the signal transduction. Techniques like molecular dynamic stimulation are used to predict how protein behave over the time at atomic resolution. Now let's talk about the protein ligament interaction. Protein interaction with other molecule based on the physical principle. The binding process involves the force like electrostatic van der Waals van der Waals interaction and hydrophobic force. The strength of this interaction is measured by the binding affinity, which can be influenced by the protein structure and the environment. Now let's discuss about the goal of protein physics. Understanding protein folding. Predict how the protein will fold based on the atomic amino acid sequencing, the protein folding problem. Determine the principle governing the thermodynamic static of the folding protein. Modeling protein dynamic. Develop accurate models to simulate the dynamic behavior of the protein in their natural environment. Understanding how the molecule motion release to the protein function, such as how enzyme catalyze reaction. Protein structure function relationship. Lithocyte the relationship between the structure of protein and their biological function. Identify how the small change in the structure due to the mutation and the environmental charges affecting function. Predicting protein and protein ligament interaction. Predicting how protein interaction with other molecule or ligament, which is critical for understanding biological pathway and drug design. Use technique like docking stimulation to understand this interaction at the atomic level. Developing therapeutics. Understanding the physical basis of the protein misfolding diseases to create a treatment that can predict or reveal this misfolding process. Design molecule that can modulate protein function through previous interaction mechanism. Biophysics method development. Important techniques such as X-ray crystallization and MRI spectroscopy to detect the protein structure with a great prediction. Now let's talk about the application of the protein physics. First, drugs discovery. Understanding the protein ligand interaction help in designing drug that can be targeted specific protein involving in a disease process. Biotechnique. Protein engineering relieve to understand the physical property of a protein to create a protein with a new or important function for industrial or the medical use. Understanding disease. It can be used in protein physics, help explore the molecular basis of the disease caused by the protein misfolding, aggregation, and mutation leading to a targeted treatment. Sympathetic biology. Designing new protein or modifying examine one to use knowledge of their physiological property, especially in the sympathetic biology, for creating new biological system. Thank you.